very much for having me. It's very nice to see you all. So I'm at Fred Hutch. I just like my history briefly is that I started there as a postdoc in 2015. I did a PhD in physics. So I had a lot of biology to learn over the years, which some of the people in the room have taught me a bunch. So thank you to them. Yeah, and now I'm a staff scientist at the Hutch, and I'm now an affiliate with Global Health. Also thanks to Grace and some others. So yeah, it's really nice to be here and meet some of you in person. And to everyone online, hello. So I guess I'll probably talk to you, so we'll see. You'll see the slides. And I did kind of put together like a sort of I think there's 3 or 4 like mini sections of the talk, and I guess if we want to go faster through anything, slower through anything, we don't have to get through it all, but we'll see. So I want to tell you about my work over the last few years, and I guess honestly before the pandemic, then we did a bunch of COVID work, and now we've been doing HIV work again. So there's a little bit of a hiatus in there, but the title, Passenger Hypothesis, is thinking about how HIV persists during suppressive antiretroviral therapy in people living with HIV and the sort of biology, immunology underpinning that. Okay. Okay, so I'd like to start with the acknowledgements, just so that doesn't get washed away at the end. But I'm really grateful to Josh Schiffer, who's my postdoc mentor and continues to be a mentor, and many people in his group. Elizabeth Duke in particular taught me a ton of HIV biology back in the day. Fabian, who has been working with us for a while, taught me a lot about modeling, and some other folks at the Hutch. The sort of next couple panels this is sort of the sections of the talk. There's collaborations now that I have with groups at UCSF, at Hopkins, at Rockefeller, and now with some of you in Global Health. So the story sort of breaks down by collaborative work. And I always say I'm so grateful for the people who actually give their bodies to these studies. It's pretty unbelievable that some people will do that, and we learn so much from them. Okay, so motivation-wise, I was able to find this photo from 2017. I think this was one of these cell and gene therapy conferences, but this is Timothy Ray Brown presenting to Fred Hutch, and he was sort of the Berlin patient, if you've heard of that, or one of the first people who was either cured of HIV or was in long-term HIV remission, depending how optimistic you are. But he gave a really wonderful presentation. It was like this, he understood the science like down pat and had this very humanistic perspective. It was a really wonderful thing. So this was one early introduction for me in thinking about HIV cure. And I think that's sort of motivated me through the years, is that there are these interesting cases of people who appear to be cured of HIV. And I think the more we learn about them and the more we learn about this system, possibility of making it happen more. I often also say that, at least I wasn't trained in medicine, but to me, antiretroviral therapy for HIV seems like one of the greatest successes of modern medicine, or goes from a sort of death sentence in 10 years to being a very manageable disease if you live in a place and have the resources to take medicine. So this is a curve of what happens when people start taking highly active antiretroviral therapy. So there's viral load set point from HIV, and then it goes away pretty rapidly in several phases. However, unfortunately, if the drugs are stopped at any point, in most people, the virus rebounds. And so what we are interested in is this sort of undetectable period, yet not going away completely, because when you stop taking ART, the virus comes back. So this is the HIV reservoir, sort of conceptually, and we'll talk more about that. And over the, so I guess now 20 years ago or more, people have basically figured out that what is persisting are latently infected CD4 T cells that have HIV proviruses integrated into them. And then when drugs are stopped, they can reactivate the virus. And unfortunately, if you go measure these latently infected cells, you find that they go away extremely slowly over time in people living with HIV for this, in this case, 7 years. There's a 44-month half-life. So a sort of natural cure, if you want to think of it that way, would take 70 years, 100 years, if this even continues. And we're obviously, you can't be sure of that.
Are those cells replicating with the virus in them, or are they just really just living there for 7?
Great question.
7 years old.
So I love that. And okay, so this is the passenger hypothesis, or at some level. No, I mean, I think that's very well said. The question for me is, like, what what could possibly be going on in this passenger hypothesis is that the latent HIV proviruses are following basically the processes that normal CD4 T cells do. So that's division, death, and I'll tell you about some subtleties of that. So I don't think, short answer, that it's just cells living a long time. I think they must be dividing and carrying the provirus on with them. So yeah, but not viral replication. Okay, so the things that I think about in latently infected cells are, these are CD4 T cells, but and this sort of helps you just frame the rest of the sections of the talk, but there are different types of CD4 T cells that do different things in the body, and I'm not an immunologist, so you all probably know more about this than I do, but for example, there are naive T cells and then memory T cells that do different functions in the immune system. Then each of these CD4 T cells has a T cell receptor, which tells it what to be specific for and sort of which pathogen to fight. And then in a latently infected cell, there is an HIV genome integrated into the human genome, and that can tell you something just about, I guess, like, yeah, what is that sequence at first, and then also how well does that sequence work, i.e. is it an intact sequence? A lot of integrated HIV proviruses are defective and actually can't replicate. So to understand this previously, and that this is now almost 4 or 5 years ago, we developed this idea of thinking about HIV provirus sequences as a sort of ecological species. So what you find when you look into HIV sequences in people living with HIV is that there are identical sequences. And this is basically impossible by HIV replication because it mutates so much. So this was some of the first evidence that these cells must be dividing, and therefore the cell process replicates the sequence rather than HIV virus replicating the sequence because it makes too many mistakes. So this is a cartoon of a phylogenetic tree with identical sequences. And in this case, we sort of got rid of the tree and we thought about it instead as like ecology, where there's this many of the yellow species, this many of the green species, this many of the blue species, and the distributions of those abundances tells you, or you can learn about the underlying processes that generate those abundance distributions. And we took some stuff from animal ecology, like, you find 8 wolves and 2 foxes and whatever in the forest, and you can learn about how many species there might actually be, and or what is the competition between them and so forth. So I'll refer to these as rank abundance distributions. So it's always ranked from biggest, so like most common species to least common species, left to right. And then the y-axis is the abundance of those species. So the first finding, I'm going through this quickly, but that's because it's from a while ago now, but these are data from Thor Wagner, who's actually at UW, or maybe Seattle Children's at the time, and Frank Maldarelli. So there's these 2 science papers that came out in 2014 that show the clonality of the HIV sequences. And then this presentation of it and sort of thinking about it in this rank abundance way is from our paper, and we modeled these distributions and sort of extrapolated them. It's really hard to find enough HIV infected cells, but you can extrapolate these rank abundance distributions. And what's interesting is, despite when you look at these rank abundance distributions, for example, there's these flat lines at the end, and these are just all sequences that were only found once. But I think because of how many infected cells there actually are in the body, those are not truly singleton sequences. What they actually are is sequences that are probably clonal, but less clonal than the biggest ones, but just happen to be sampled once. So we used to call this the iceberg, or something like that. You're only looking at the tip of the iceberg here. But when you do that extrapolation, what you can kind of infer is that really most HIV proviruses are clonal. So like more than 99% of these HIV proviruses are probably clonal, and therefore kind of have to originate from proliferation of infected cells rather than viral replication. Some corollary things that you can come up with from that too is that if you estimate how many infected cells there actually are in someone's body, which is a little bit hokey, I would say, but like possible, the largest clonotypes might have 1 to 1 million associated infected cells. So those would be huge clones. And if you keep going with this kind of calculation, napkin calculation, that would require 20 to 26 cell divisions, which I don't know really how this works, but ultimately there is some limit to how many times cells can divide, this Hayflick limit, if you've heard of that. So I think this is getting near that. And I guess it would be suggesting that these infected cells are the CD4 T cells that respond to common antigens. That's why they proliferate so much. And yeah, like cytomegalovirus, for example, is a candidate for one of those antigens. Okay, in the next work going on from here, I then started working with Annie Antar at Hopkins, and she had done this study where they looked at HIV clones at 2 different time points in the same people. And the rank abundance distributions are here. And I think what you can see a little bit by eye is that they start to become a little bit more clonal at the second time point. And what I'm looking for is just that there's the biggest clones have like 10 cells rather than just like 2 or 3 cells. And then there's a little bit more structure here. So the idea here would be that there's not actually new HIV diversity being added to the system. Those cells just keep proliferating, and therefore they get more and more clonal. And then, yeah, more modeling type projections is that you can say that maybe the top 10 clonotypes actually have 15% of all the cells or something like that. So it's a really dominant system by the big clonotypes, or the top 100, or maybe 20, 25%, the top 1,000, maybe a whole 30%. And then over time, these numbers can increase to where the top 1,000 clonotypes might have 60% or something like that of all the cells in the body.
Yeah, doesn't that mean that over time, because we can only sequence so deeply, if you sequence the same number of proviruses in T1 and T2, it would look like the diversity was actually going down. Do you think that's true? Like if that's what the data would look like? Because your clones are getting bigger, so like the number of cells you're getting for each one is getting more higher abundance, and so you're not able to see the diversity even if it's there.
Yeah, that's
So that
well said.
you would see the diversity. It would look like diversity was decreasing.
It's still hard to tell. Like if you do sort of statistics on these distributions, like a little bit by eye, to me, it looks like this is getting more clonal, but depending which metric you choose, it's so I think this idea of looking at, I had drawn that in the cartoon before, but basically looking at the slope of this distribution seems to be a pretty useful metric that summarizes it even when it's really undersampled. But yeah, that's a problem that the oncologists have been working on for a long time. It's like, how do you actually estimate the diversity when you only have this tiny subsample?
Yeah, and really quick for this one, what's T1 and T2?
Oh, thank you. T1 is like 10 months to 12 months after initiation of therapy, and T2 is like 7 to 9 years.
Oh, wow.
So they're actually pretty far apart. And I'm thank you for bringing that up. This will come up again, and I think I will have the numbers on it. So these same people we're using in the next study as well.
They're adults.
Yes, they are adults. Oh, great. Here it is. Okay, so this is Annie, and she's done a fantastic amount of work to enroll these people and follow them. They're extremely well ART suppressed, and so there's 4 people living with HIV. Those are the data I just showed you, and they acquired HIV at least a year before starting ART. That's harder to know. Acquired HIV, and then the first time point is at T0. The second time point, this is the T1 that we were just looking at. So I misspoke. I said 10 months, but 2 years. And then, so 10 months is the 0, 1, 2 years, and then 9 years is like the approximate range. And then we're interested in studying the clonality of the CD4 T cells at the same time in these people. So now we've, like that last slide was about the HIV proviruses. And then now we're interested in just the clonality of the T cells. And so she also found this group of age, race, and sex matched people who are HIV negative, but have the same sampling times. So this is not HIV sequences at this point, but it's CD4 T cells just to sort of compare among people with living with HIV and not. And then I don't think I'll really show you much about this, but there's also some published cohorts of people now, like including this one that has 33 humans in it that have adults that have CD4 T cells receptor sequencing done, so we can sort of add to our sample size, compare and contrast with some caveats, obviously. Okay, so from those people, from the people living with HIV in particular, she isolated the resting memory CD4 T cells, and then split that group up. And on some of them did this TCR beta sequencing, actually at this company, Adaptive Biotech, which is in Seattle, and is a Fred Hutch spinoff, I learned recently. So we send, you send the cells off to them, they do the sequencing, and then you can kind of compare and contrast with the HIV infected cell sequencing. And this little like fork here is maybe a little bit important because it's just to note that it's not exactly the same cells. So you would need like a single cell sequencing approach to look at the sequence and the TCR receptor from the exact same cell. This is more of a holistic comparison that we can make.
How much does that sequencing cost?
It is about $1,000 per sample. And so here, I guess that's 4 people, 3 samples each. So yeah, 12.
That's the TCR sequencing.
That's right.
And then the HIV sequencing is on top of that.
Yeah, and we can talk about the.
No, I was just curious about the T-cells.
Yeah, we have the data from some of the kids now. Okay, so the left panel, or sorry, I guess the punchline of this slide is actually, surprisingly, HIV is more clonal than CD4 T cells. So the passenger hypothesis idea, I would have thought that their clonality, like my hypothesis going into it was going to match, which would have said like, oh, the processes generating the HIV is the same process generating the T cells, or some inference like that. But when you actually look at the CD4 T cells, this is a rank abundance curve for tons of different people, including those extra cohorts. And I think a few things to observe from this is, first, there's way more cells that you can sample when you're looking for CD4 T cells versus HIV proviruses. So you get a much better picture of the whole distribution. So it goes out to tens of thousands of cells. And then there's a pretty big range in the sort of how big the biggest clone is. So some people's biggest clone is 1 in 1,000 cells, and some people's biggest clone is like 1 in 10 cells. So really abundant. And to make a fair comparison against the HIV sequences, we have to do a computational resampling. So you take this distribution and assume this is kind of the ground truth. And then you say, what if we looked at this distribution, but we only grabbed 100 sequences, which is sort of similar in sample size to the HIV proviruses. And you get distributions that look like this. So there are some things that you find 10 times, but a lot of times you just find things twice. And then a lot of times you just find these like singles over and over again. Whereas doing that for the HIV proviruses, you do find the red lines are the things I showed you before. The orange is like a new cohort that we sort of filled in this with. But you do find much bigger clones in the HIV proviruses. So there's some discrepancy here. Maybe things are a little bit different.
Dan, your orange and your red seem to not be so overlapping. So is there some bias in your cohort?
Great point. And then you know the distinction here, but the red ones are total HIV DNA, and the orange ones are ones that grew out from QVOA. So in the paper, we're not including the orange anymore. We're just comparing red to red, and it's a little bit less obvious by eye, but I think the stats still hold up.
What is QVOA?
Quantitative viral outgrowth assay. So this is a pretty time-consuming assay, as I've been told, in which they try to actually grow virus out of these provirus or out of these lately infected cells. And it means, or what you find is that most of those cells that you find HIV DNA in still don't make virus. They have some sort of defect in them, or are no longer intact.
The other thing about that assay is it's not necessarily reproducible. Like each time you would get different, like it's basically like there's some stochasticity to the reactivation. So what you reactivate the first time you did it in the same set of cells is different. So there's potentially some bias in what grew out.
Great point.
But if you only had the red, would
Yeah.
it look more similar to the
So it seems like
sample?
Yeah, a little bit, and I'm sorry I didn't put the proper box plots, but it is, at least by some of these ecological statistics, it's still
Different.
yeah.
And then why was your hypothesis that it would be the same clonality when the HIV infected cells are just a fewer subset than the whole CD4 population? So to me, it would seem that that subset would have a limitation just because it doesn't infect all your cells.
Mm-hmm.
Whereas when you're looking at all your memory T cells, they're looking at everything. So there should be a lot more diversity there.
I guess I was thinking of it as like during active viremia, the cells that get picked to get infected, they don't choose to get infected.
Yeah.
They or maybe they do have some
Propensity,
yeah,
yeah.
like propensity, yeah, which is probably that they're specific for HIV or something. So it's sort of uniform seeding of the whole distribution. And then therefore, if you look at the, like, you know, if you sort of drop HIV sequences through the big distribution or something like that, and then you resample again from that to get to just 100 of those cells, I was thinking that the distribution would still remain, remain constant with that idea of if the uniform seeding is real. So that's probably one of the possibilities for why they don't look similar, is that may not be the right hypothesis. We've mostly looked at after ART rather than like what happens before, but I think I might get to that in a little more detail in 2 slides. This is now, so this comparison was sort of like just these clonal distributions, how similar are they? This is another comparison that we can do, which is another thing that turns out is different between CD4 T cells and HIV proviruses, which is that HIV DNA tends to decay over time in people, whereas TCR clones do not. And this is, I guess, these people living with HIV looking at HIV DNA from the IPDA assay, which I'll talk a little bit about, that differentiates between intact and defective sequences. And these intact ones tend to go down over time, and the defective ones kind of tend to stay flat. And then this is a graph of just 1,000 clonotypes from one of these people, tracking them over these 3 time points. So now that you have these TCR clonotypes, you can see some of the ones that have lasted in these people. And if you kind of make a model of this line, some of them go up, some go down, but the general trend is basically flat. So you're left with this sort of rate of change where both, sorry, the colors don't match it, or I guess both in the TCRs, in the people living with HIV and the HIV negative controls, the TCRs don't really go down. They, you know, depends on which clone you look at, but the average doesn't really go down, whereas the intact clonotypes do tend to go down with, again, sort of like this 40 to 60 month half-life that has been observed a bunch of times before. So there's some discrepancy here as well, where HIV DNA decays and TCRs don't. So they must not be like perfect passengers in this system. And then another thing to look at is in each of these people, this is the rank abundance distribution with the 3 time points. So if there's 3 lines in each of these panels, and what's interesting is sometimes they're so overlapping, it's kind of hard to see that, which this is one of the more amazing things about this data set to me, which is that even though these clones are changing their sizes, the distribution of so sort of like the difference between the biggest to the second biggest to the third biggest to the fourth biggest or whatever, that seems to stay really constant in people over time. Like there's this hierarchy of sizes that is preserved by the system, which to me says like immune system homeostasis is like unbelievably good at able, you know, and you see some changes, and you actually do see more changes in the people with HIV than the people without HIV. So maybe the system is a little bit out of whack, but not really. So this structure is staying remarkably consistent, whereas I showed you already before that the HIV clonality increases in time. So yet another kind of discrepancy here. And
Sorry,
oh yeah,
the second
yeah,
number 2, the slide before this one.
yes.
So like the intact and defective looks similar to, you know, the purple and the green, and then it's like the intact one, your last figure there is the one that has differences. And so has most of the work been intact, or you just study everything together?
In this, like in the other HIV clonality things, that was total HIV DNA. So it's not distinguished to intact and defective.
But it's predominantly defective, so it's kind of a better measure of defective than intact. Intact is a small proportion. So if they're
Very
looking
good
at
point.
total,
Yeah.
it's kind of more similar to defective.
And so that, but intact is probably more interesting
Yeah.
Yeah.
It depends on the question you're asking, but yeah.
Yeah, I think generally, yes. Because, yeah, and I didn't say this before, but with the rebound, right, like these things have to be some definition of that rebound competent, whatever that is that intact? Is it something else? But intact is a good approximation of that, I think.
And this is consistent with many other
Yeah.
people's work with the intact kids best.
Okay, so this is kind of a dense figure, but I think that this is a pretty fascinating one too. So I just showed you that those structures are like the same over time. The x-axis is the initial size of a clonotype in a person. The y-axis is how much that clonotype changed at the next time point. And then the color is how often that event was sort of observed. So what you see is purple. The most common thing is something that was pretty small and then stayed about the same. You also see stuff that was pretty small and then grew even by like a factor of 50 or something like that. And then you see things that were initially big, like 20 to 50, and then shrank. But what you don't really see is things that are big and grow. So there's this kind of, again, homeostasis system here where things that are small tend to grow and things that are big tend to shrink. And there's this like constant dynamical system buried in that where, things that are big are shrinking over time and things that are small are growing. Yet there's that persistence of that structure, again, of like the relative sizes. So there's a very dynamic system that, keeps like an equilibrium distribution, which is honestly a pretty hard system to design. If you're like an engineer, I think you'd be impressed by how stabilized the system is. Okay, so and I would say it's been shown that HIV proviruses do also tend to grow and shrink over time. And so perhaps that is a little bit of a passenger dynamic, or it's not inconsistent with a passenger setup. Okay, so the next, for me then, I was interested in, can we model these dynamics mathematically, computationally? So we built a little mathematical model that has some pretty simple rules, which are that there's a bunch of different T cell types, like each of these colored circles, and then some are bigger than others. And then some of the T cell types are carrying HIV proviruses, which are these little pentagons. And then some of those proviruses are intact, which is little X's. So sort of like the levels of subsetting here. And the rules of the system are that the T cells proliferate and die, basically in balance, but sort of stochastically. And TCRs, or sorry, new clonotypes. There is the ability to have new TCR clonotypes. Your immune system does get new diversity over time, I think less as you age, but what's different about that is there's no introduction of new HIV diversity because there's suppressive ART. So the model, like this is just like a little fun example of the model, but imagine you have each of these TCR clones and you just sort of run the system. Just by stochasticity, you get some that die out and some that grow big, and you can kind of get this this waxing and waning to happen on its own. And when you actually look at some trajectories of the subsetting, this is just examples of things that could happen. These are months, so almost 10 years of ART, and cells in each of these clonotypes, where the yellow ones are the TCRs and the purple things are HIV proviruses. What you can see is there's things that happen where, maybe there's just a cell that has no HIV in it, or maybe there's a cell type that actually started small and had HIV in it, but then proliferated a bunch. So most of the cells in that clonotype now have HIV in them, and you can get a bunch of different possibilities here. Trying to match that to data is tricky. But the simulated output here, this is just one example. You can generate a system where basically the T cell level stays the same. The intact HIV DNA goes down precipitously. The TCR rank abundances kind of look like the ones that we looked like in the data. And then the HIV rank abundances also kind of look like that and kind of get more clonal over time. So just approximating the data here, you can kind of do that with this simplified version. However, one important thing that you need is just that the HIV infected cells need to die faster than the general CD4 T cells. And I don't think that would surprise people in the field, but it is, yeah, kind of nice to see that perhaps there's a little bit of, you could say, a negative selection in the system against HIV infected cells versus normal T cells. Okay, so that's the result of the modeling. The summary here is, I would say HIV is not completely an inert passenger on the CD4 T cell dynamics. And then the summary here is that the decay rate of HIV is non-zero. It seems to go down over time, whereas TCRs basically stay the same. HIV clonality is a little bit higher than CD4 T cells, and HIV clonality seems to increase over time, whereas TCR clonality seems to stay about the same. Yeah, so maybe HIV infected cells are under one or more selective forces unique to carrying a provirus. The provirus might change the fate of the cell. And that's, there's been a bunch of really nice papers in the last few years that are showing similar things on like a single cell level.
Are they presenting anything on the outside of the cells?
I sort of forget which if any markers. It's not clean. Like it's not like there's some surface marker that says I'm an HIV latently infected cell, but maybe if you do some
Nice, then you can.
I mean it's
That's what everybody's trying to.
yeah.
I'm just right, the thought is that they must have something out there that.
Yeah, that makes
has
sense.
other
Yeah,
cells.
eaten up, yeah.
Yeah, you're
Or they die.
Or it's that they're expressing virus and like putting themselves off. I mean, it can, there's
If they
many things
suddenly
they could be.
Yeah.
Start
A
to.
little
They
bit.
do
Wake up.
express, all of a sudden they'll get
Mhm.
activated and express and then they.
yeah, and then maybe they get recognized.
Yeah. Get overeager.
Mhm. Okay, so that's one little recent story. This stuff's not published yet, but we're trying to finish the paper. And now I want to tell you a story about working with, this is Charlene Backus-Sufan and Peter Hunt, who are at UCSF, and or Charlene's now in the industry. And she did a ridiculous amount of work tracking 37 people living with HIV and looking at those CD4 T cell subsets in the HIV DNA in those subsets in all of these people. So this slide is just to show you that this is a pretty heterogeneous group of people, although they are 37 men, basically from San Francisco. So it's not a heterogeneous global population, but in terms of clinical history, there's a range of time on suppressive ART and different sort of total reservoir sizes. So the y-axis here is total HIV DNA. And then the color is time between HIV diagnosis and ART initiation. They know that somewhat. So there's a mix of these variables in this system, which may be useful to talk about. It also may just make the analysis harder. So yeah, Charlene sorted HIV DNA into 5 CD4 T cell subsets. These are the acronyms at the bottom. So there's naive, stem cell memory, central memory, transitional memory, and effector memory. And I think the immunologists have very well-defined pictures of what each of these subtypes do and what markers they express and so forth. For me, the simplified version of this is that it's sort of a maturation pathway from being a naive cell to being the most capable of attacking the antigen at the end, the effector version. And the HIV DNA in each of these subsets, again, is really heterogeneous. The like dots and lines are the individual trajectories, and then the flat line is our best model through the middle of all of that. So it's hard to come up with like an average, or I guess you can come up with an average, but you have to think about the heterogeneity. And the other amazing thing that they did in these people is they did a heavy water labeling experiment to look at CD4 T cell turnover rates. So that's independent of the HIV. You can look at how fast CD4 T cells turn over by labeling them with this deuterated hydrogen, deuterium. And it's a really cool comparison here because you can look at, this is the sort of, each of the dots on this left panel is the decay rate of the HIV in one of those people. And then each of the dots on the right is the CD4 T cell turnover rate in these same people at the same time. And what it looks like is that maybe transitional memory and effector memory cells tend to lose HIV DNA over time, whereas these other subsets, HIV DNA is pretty constant. And meanwhile, there's tons of proliferation in these subsets. So they are like this, the CD4 T cells are growing and dying. And the picture I have of that then is that, for example, in effector memory, you get lots of cellular turnover events at 2 different time points, but some small fraction of those turnover events lead to HIV clearance. So the HIV DNA as a fraction goes down a little bit over time slowly, even though the total number of cells stays about the same. And I guess previously in this paper from Charlene, she observed that the DNA levels were correlated among the subsets. The first paper was just at a single time point, whereas these data are all this longitudinal study. And so that gave us a little bit of motivation to try to model the system as this connected thing between the subsets. And so here I'm saying, yeah, what rules govern the system? And before I was mostly focused on this idea of proliferation and death in that model I showed you before, but I think now that you have this broken up into these subsets, you can actually think about, do you need to have this cell maturation pathway? And is it possible that HIV DNA gets taken along for that ride as a cell matures in its phenotype? So we fit a bunch of different models, these longitudinal data, and tried to come up with the best model for this. And I'm gonna show you this crazy movie we made to try to explain this. Actually, sorry, we did not make this. We gave it to an animation company because I couldn't figure out how to make this. So what you see here is there's the 5 subsets, and this is our best estimate of the rates of these things happening, which is anytime you see dots flowing from one subset to another, that would be cell differentiation, carrying HIV DNA. And then the stuff falling off the bottom is death. And then the flashing is proliferation. So you have all 3 of those things at once. And the reason that there keeps being more dots is that it's broken up into different time scales. So now it's going to look pretty crazy because this is like what might happen in a year, how much cell like differentiation happens. And I think what you can see is that there are certain subtypes that are really proliferating a lot and really dying a lot. And that's, yeah, based on those heavy water estimates of how much this stuff is happening. So there's like a lot of flow basically from the central memory cells onward. And then there's a lot of proliferation in the memory cell compartments. And then there's kind of less stuff happening in the naive and stem cell memory. But this is, yeah, I guess our best estimate of what really is happening in this system, which is, again, a very dynamic system. And I think the sort of summary is that it might be that a lot of proliferation happens in central memory, and then that gets flowed on to the other phenotypes, and then the effector memory cells at the end have to be dying really fast to sort of keep the system all in equilibrium. I do have this like much easier to look at summary picture, maybe less fun to look at.
That was cool.
Great. I hope it helps. I don't know if it's too much, but yeah. So the summary here is then like averaged across all the different people. If you try to calculate how many times in a given year these different things happen, what you see is like maybe there's like 10 naive proliferations, then maybe like 11 or something naive deaths. So naive differentiations, and then a few less naive deaths, so they stay about the same. In the central memory compartment, the circle is, there's a lot of proliferation happening and maybe some onward differentiation and a good amount of death. And then the orange and yellow, the sort of more differentiated memory subsets have some proliferation, but a lot of death to sort of balance everything out and keep things from going away too fast. So again, the heterogeneity is huge between all the people, but if you try to summarize it as averages, this is what it looks like. Okay, so I would say that's the mechanistic contributions to the HIV persistence. Again, sorry for your comment before, that's for total HIV DNA, so not necessarily intact. Maybe that has different rules, but we just don't have those data. And because we built this complicated model, we can now play around with what happens if we do some sort of, I guess we were calling it in silico knockout experiment. So you do some computational thing where you drop one of these mechanisms. So this is the picture of the system on the left with the four things that can happen: the differentiations, the proliferation, and the death. And then the subsets stay about the same. And this is like some hypothetical study where you measure at 0 years and 3 years. And the idea would be you don't really see difference between these box plots because not much changes for the total HIV DNA. If you do two different theoretical therapies, you can start to imagine, or you can start to understand why, but also see that maybe the levels would drop over time in this 3-year interval. The first thing we suggest is something called anti-proliferative therapy, which would be just blocking proliferation at some level. In this case, the example is just reducing the proliferation rate by a factor of 2. And in particular, you see that those subsets that proliferate a lot are very different when you block proliferation, because assuming they still die at the same rate, they're not getting created infected cells. So maybe this would mechanistically be a hypothesis for how to reduce HIV proviruses. The other thing that we simulated was what happens if you enhance differentiation. And for both of these things, there's probably candidate molecules you can think about to do these things. But if you enhance differentiation, it sort of pushes the HIV proviruses down through the maturation pathway to those cells that seem to die more. And that's maybe another way to reduce proviruses. So this is very theoretical. This is just simulations, but I think a good thought experiment for once you understand the mechanisms, then can you leverage those mechanisms to reduce HIV? Okay, so summary there. HIV DNA persists, clears slightly differently in the different CD4 T cell subsets. The proliferation is the dominant mechanism overall, but differentiation might play a role in carrying HIV throughout the system. And the same mechanisms that sustain could be leveraged to reduce. So yeah, okay, good. I have this caveat here. This is total HIV DNA, may not be rebound competent. So we might be learning about something related, but not perfect. Okay, there's 15 minutes left. I sort of feel like I should just skip through this next thing. This is about assays, and Dara, I should tell you about it sometime. But yeah, let me get to this. So yeah, now thanks to some of you all, I've been thinking about the similarities and differences of these systems in a pediatric setting, which I think of as a vulnerable but potentially powerful system for two reasons: because they have low reservoirs from early ART initiation, and it's really interesting to me if we can figure out any immunological advantages that cohort has over the adult cohorts that we've been looking at. Okay, so this is the sort of crazy raw data that, yeah, Jill, you've looked at a lot, I think. HIV RNA in 260 kids from Nairobi over years and years of therapy. Some of them suppress right away, some of them don't, some of them suppress later. CD4 T cell counts from all those, or a lot or all of those children, and then intact and defective HIV DNA in those same children. So a massive effort from you all working on this cohort, and I think one of the best data sets, that I've ever seen as far as detail and numbers.
What were the kids getting? This was OPH, was this?
Yeah. Do you mean?
American?
Yeah.
Just because
A lot of them
the
got
kids, it doesn't look
healed,
like they're suppressing very well.
They don't
It's
suppress
kind of
well.
sad. But kids should, right, with
They don't.
GGG or no?
Yeah, now they will, but
Yeah.
infants still have a hard time.
They were enrolled in like 2007.
Okay, so it's
2010,
before
so it's a long
some of them
time.
were potent.
And kids just generally suppress a lot worse than adults.
They have higher viral loads.
Yeah, and also, therapeutic doesn't taste good, or, they might have resistance.
Mums, not all mums were on ART, like for guidelines. So it's also tended to have higher RNA levels.
So I think the next slide is subsetting to those that did suppress within a year. I just wanted to look at basically the subset of all those lines among the ones that actually did go down. And I think, yeah, we need to have more conversations about what kind of biases that would even introduce by doing that. That's not something I've thought about much. So very preliminary look at these dynamics is that before 1 year, at least among these kids who suppress within a year, the suppression is really fast. So if they're sort of destined to suppress, then it goes down within the first few months. And accompanying that is a general rise in the CD4 T cells. And I think what's kind of cool about this system is that you would usually expect that in adults as well, as these cells that are getting killed by HIV are now not getting killed by HIV, but also the CD4 compartment is changing in these kids simultaneously. So it's kind of a interesting convoluted problem that I think modeling may help with. And then in that year, there's also a pretty good decay of intact HIV DNA and a slight decay in defective HIV DNA. And then after 1 year, this is the sort of RNA status. So you can't estimate a decay from that. It's just flat. But the point is there is some viral blipping happening, but it stays pretty low in all these people. And in that realm, now CD4 count starts to decline a little bit. And this, I think, would be a component of the system, which is immunological development. So after a year old. Whereas intact HIV DNA tends to go down, but not as steeply, and defective HIV DNA tends to flatten out. So the summary of these rates is that within the first year, there's a really rapid viral RNA clearance. And then after that, it flattens out. Oh, just this is all the little decay rates. They're colored the same. The dot's the average, the line is the confidence interval, and then the arrow just means it's like a confidence interval that goes up above the scale. So yeah, very short half-life of viral RNA in the first year, but then flat. CD4 T cell count is basically flat in the first year and then starts to decline a little bit. Intact HIV DNA is going down, and then after a year is actually remarkably consistent with the best estimates from adults. So this horizontal gray line is a number that's been published in the literature as the decay rate of adults. So it's kind of incredible that the circle's lying on it. So maybe it's about the same in kids, or maybe that's just there's a big confidence interval. And one of the things that we were interested in when we started this project is whether or not the intact decays faster than the defective in kids. And sort of the mechanism behind that, I think we've already talked about a little bit, is like maybe intact HIV DNA can express itself or do something that the immune system targets and kills more so than the defective HIV DNA. And so at least we're sort of seeing that same phenomenon in children. It's not, there's not some extra thing that looks like it's happening here. That story remains basically consistent. And then the last thing per that sort of motivation slide is that, this is another one of these complicated hexagon plots. So let me talk you through that first, which is the x-axis is time, the y-axis is intact HIV DNA. So you do see that this goes down over time. But then the coloring is the ratio of intact to defective. So right at the beginning, you see much more yellow, which basically implies that when there's viral load, most of the sequences that you find are intact, which sort of makes sense. There's this active replication happening. And then later in time, because of this, we say differential decay, the intact goes away faster than the defective, you start to see much lower ratios, much more blues and purples after years of therapy. But I think the other interesting thing about this is that there are kids in this cohort that have a very low ratio of intact to defective HIV proviruses right at the time of initiating ART, which is a very nice setting for avoiding rebound if you already have much lower intact proviruses. So those people also have they have the lowest number of intact HIV DNA, but that doesn't mean that they have the absolute lowest number of defective sequences. They just have a slightly lower ratio of intact to defective. So I think that's more sort of encouraging evidence that this setting is one to look for remission and really work on as a population that could be treated. Okay, this is the summary. Persists due to proliferation. Like if I can give you some takeaways, it does seem like that's really the thing that's sustaining HIV proviruses during therapy. It's probably not an inert passenger, especially in certain subsets. It seems to be influencing the fate of the cells. DNA appears to be passaged through the subsets through physiological differentiation, just normal CD4 T cell maturation. We might attempt to block those mechanisms now that we've learned about them. And pediatric cohorts are understudied, not by you all, and may be particularly amenable to HIV remission. And more insight on the immunological mechanisms, hopefully coming in the future. And we've done the TCR sequencing to look at the CD4 T cells as well in like 8 of these kids now. So I'll be excited to show that someday. Okay, I'll just end on this. And yeah, thank you all for your time.